Next up, we have oral presentation 5, Concord, North Carolina, Mission 11 Flight Experiment.
Good morning, I'm Paige.
And I'm Zainab, and we are from Concord, North Carolina. Unfortunately, our other member was not able to make it today, but we are still very grateful that we can present this morning. We are presenting to you our presentation about the gravitropism of radish seeds in microgravity.
In the 2016-2017 school year, our school participated in SSEP, where all the students in the STEM program wrote proposals for an experiment that could be performed in microgravity. Our proposal was selected for our school to be sent aboard the International Space Station. It was sent to the ISS in August of 2017 and was sent back to us for harvest and analysis in September.
Our main question was, how does gravitropism vary in different environments, the environments being in space and on Earth? Gravitropism is the focus of our entire experiment, and it simply means how the plant responds to the stimuli of gravity. Since these two places have different amounts of gravitational pull, we thought it would be interesting to test plant growth.
The purpose of our experiment is to find out if a seed can germinate properly outside of Earth's gravity. Germination is the first stage of a plant's growth, so it is extremely important that this happens properly. If the seed does not germinate, the plant will not grow. By doing this project, we learn more about agriculture in space, which will benefit astronauts in many ways, being used for medicinal, eating purposes, or even growing plants on different planets like Mars. If the gravity in space is not suitable for plant growth, we need to find a way to modify the plant or the environment in order to grow crops in space.
Gravitropism, as mentioned before, is an important part in plant development. There are special organelles in plants that cause gravitropism. In summary, a type of plastid in plant cells called amyloplasts settles to the bottom of a plant cell due to gravity. When the amyloplasts come in contact with a network called the endoplasmic reticulum, calcium ions are released, triggering an indoleic acid, a type of auxin, to fall to the bottom of the cell. Gravitropism is what causes plants to always grow vertically rather than drooping. The IAA stimulates growth in the shoot so that it can grow against gravity, which is called negative gravitropism. It also slows growth in the shoot so that it can grow along with gravity, which is called positive gravitropism. Vertical growth is vital because it prevents the fruits and flowers of a plant from falling and helps it to maintain stability.
Since there is less gravity in space, we hypothesized that when the seeds germinate in space, they would have different proportions relative to their earthly counterparts. We thought the hypocotyls would straighten out earlier and cotyledons would emerge faster due to the reduced gravity, and the primary roots would be shorter than the roots of the seedlings on Earth, while the stem may grow out even longer. We also believe that the seedlings would have roots and shoots that grow in various directions rather than vertically because the amyloplasts that trigger gravitropism would not settle in the first place. Our experiment aboard the ISS was a Type 3 FME mini-lab. Our project contained 3 compartments. The first one contained 3.4 milliliters of water. The water was the activator for the plants to germinate. The second one held 2.5 milliliters of soil and 3 Raphanus savidia seeds, or more commonly known as radish seeds. Lastly, the third one held 2.5 milliliters of formalin. Formalin was the preservative that stopped the seeds from germinating so we could observe them when they got back to Earth.
Our experiment only had a couple of interactions with the astronauts. When the experiment first arrived at the ISS, they were to unclamp clamp A and shake for 20 seconds. This released water into the chamber with seeds, activating the germination process. Then, 2 weeks before the experiments undocked, the astronauts were to unclamp clamp B and shake vigorously for 20 seconds. This released the formalin component into the tube, freezing and preserving the process.
After our harvest and analysis on September 26, 2017, we observed that the lengths and visible characteristics of all germinated seeds were all very similar. Due to the fact that our results on Earth and in space were extremely similar, we have concluded that germination is too early of a stage to determine differences in plant growth in gravity and microgravity. The radish seeds develop quickly. The amount of time in which our experiment was conducted may not have been enough time for gravitropism to fully impact development. This, along with space restrictions in the FME mini-lab tube, and only being able to have one trial for the official experiment may have hindered our results, but nonetheless, we can conclude that with this data that germination is not hindered by microgravity in radish seeds.
This project has allowed us to deeply explore plant organelles and their functions, various tropisms, and how these come together in the frontier of space exploration, and we are very appreciative for this opportunity. We would like to give a big thank you to these institutions who made our experiment's flight possible, and for that we are extremely grateful.
Finally, thank you to our parents, to Mrs. McNutt, our coach throughout this entire once-in-a-lifetime opportunity, and our school for coordinating and making this entire opportunity possible. It is an experience we will never forget.
All right. Hello, I am Augustus Tirico again, and I have another two-part question for you. Firstly, you said that the lack of gravitropism might lead to a longer stem length, right? And if we're talking about farming, the stem produces the fruit or edible component of a plant. So is that the main implication of your experiment, higher yields in space?
Yes, in a way. We were trying to find out like if it would grow like longer, so the fruit would grow like. But we were focusing on germination, which is the part when the plant comes out of the seed, which is like the most important part.
Okay, gotcha. So exactly, since germination wasn't a sufficient time frame for observation, how long would you suggest your experiment should stay in space to make proper observations and collect data?
We would say maybe, since we had around 2 weeks to a month in space, we might double or triple the time period just to give it, or do multiple trials to see what time frame suits best.
Okay, thanks.
Hi, Tim Livengood, NCSE. So NASA and the European Space Agency are both developing something called the Deep Space Gateway, which will be a crewed facility that would be orbiting out beyond the orbit of the moon, with the idea that it would be occupied for a month by astronauts and then may have to persist on its own in a livable condition for 6 to 12 months before the next visit. Based on your research, both your experiment and the research you did in preparing it, do you have any advice for what you would do on having plants that would make it a more humane environment while the astronauts are there, or for actually growing food?
Maybe instead of just growing in an FME tube, maybe do it like planted in soil. That way it can actually grow out to be the plant and not just a seed.
By the way, this is common to put people on the spot for a question they never dealt with before in meetings like this. Okay, thank you.
Good job, ladies. You made it through a Dr. Tim question. Okay, thank you.